So this is a brief kind of introduction to processing Oxford Nanopore sequencing data. So preparing the going from fast sort of 5 or FASTQ files, and preparing them and assembling them to create a file that's capable of being analyzed for sort of downstream purposes, such as phylogenetic trees or antimicrobial resistance, gene identification, and that kind of thing. So I'm Lindsay, and I was also helped by Professor Neil Stoker and Dr. Sylvia, so Sylvia is here, I think, on the call. Neil might join us, but they helped me prepare this. So if you have any questions at any point along the way, then please let me know. Feel free to shout up. As I say, hopefully there's much more information in the PDF that I sent around and that I will put on the Global Health Network pages, and I'll send that link. There's quite a lot to go through, so but as I say, please feel free to shout out and ask questions. So, okay, why is this not working? There we go. So I split it into 4 kind of different sections. A sort of an introduction, so an outline of what we're going to look at, an introduction very briefly into some of the common file types you might see, and some fairly basic coding concepts that are kind of related to Oxford Nanopore and sequencing data processing. This is mainly aimed, or this is aimed at Oxford Nanopore data, which is long read sequencing, but and a lot of the programs are specifically for long read sequencing, but you might find that if you're looking at Illumina data that some of this, or that the principles probably apply in the same way, but you might find that there are different programs to use. And just because I mentioned certain programs doesn't mean that there aren't others that you could also use. These are just simply ones that I have found work quite well for me. So again, the way I've written the code and the programs that I've used, they aren't necessarily the only options. Just the option that I'm showing you today. We're then going to look at how to actually process the FAST5 or FASTQ files for the downstream applications. So that's just preparing the data and QCing it using Guppy and various QC tools. I'm then going to explain excuse me, how to assemble the sequence data using some command line interfaces. And that's either assembling using a reference genome or sort of de novo assembly, which is piecing together the pieces without a reference genome. And then a very brief look at the downstream analysis, some of the downstream analysis tools that are available. So for instance, the use of online databases and also potential programs that you could use for things like phylogenetic trees and variant calling. So we'll start off with part 1. And just before you start, kind of, you might have, or hopefully you have a basic understanding of coding, but before you really, this sort of is aimed at people with already with tools. And if you're not already at this stage, I think I mentioned it already, but there's FutureLearn, this bioinformatics for biologists, which is really, really useful. And it's how I kind of got my head around Linux commands in the first place. We've also put together on the Pandora Global Health Network bioinformatics page, lots of links and resources to sites that you might find helpful. And another really useful program that really helped me kind of understand it was Epitome Labs. I might be saying that wrong, it might epi to me, but Epitome Labs, which uses a program called Docker. And there are different notebooks within Epitome, and they're really quite useful because it's kind of a step-by-step guide to how to do something, but it shows you all of the code that you need to do it, and you sort of click play and it does it for you. So it's a really nice sort of middle ground linking the two sides together. So I recommend having a go at Epitome Labs if you haven't already. Some other things to consider is the operating system that you're using. Some programs only run on Linux or the closely related Mac OS. A lot of them don't. necessarily run on Windows, but you can use the subsystem for Windows, which means you can kind of link a Linux interface on a Windows computer, which is what I'm doing, and I'll show you today. But in the PDF, it explains how to set up the subsystem, and I know a lot of people use Windows computers, so it's quite useful. Having a fairly powerful computer is also quite useful when you get to the point of assembling, and some programs require a minimum amount of RAM. So I've explained it for each program when we get down to it. Some of the earlier ones like Guppy, you only need about 8 gigs of RAM, which is something that a normal sort of basic laptop has. But once you get to assembly, it needs a bit more because it's quite a complex and data heavy program. So it's just something to consider when you're setting something up. Having a reliable power supply is also really important, because if you're processing really big files, and a lot of these files are really big, sort of, especially if you've got not a particularly powerful computer. So you will need to make sure that you've got a continuous power supply, otherwise you might have to start again. And also having a reliable internet. Whilst some of the command line programs don't necessarily need the internet to run the commands, you will need to upload information to cloud-based databases, and you also will need to download bioinformatics tools from the internet. So things like you will need to download, and also if you plan to use high-performance computing. So again, having a decent and fairly stable internet connection can be quite useful. This slide is just going to explain kind of the general, I think everybody likes the word pipeline, and the sort of steps that we're going to sort of briefly go through today. So we're going to start off by looking at how to base call. So the program Minnow, which runs the MinIron or the whichever sequencing platform that you have, should automatically base call for you. So it will turn the FAST5 squiggle files into the FASTQ ATGC files, but sometimes it can be slow and you can do it yourself and speed it up a bit. So I'll show you briefly how to do that. You might also need to demultiplex. So if you base call yourself, sometimes it will just spit out all of your FASTQ files into one big folder. And before you then move downstream, if you have used barcodes, you will need to separate them into barcode 1, barcode 2, barcode 3. So that same program can be used to do that. Code 1, you can then trim off any of the additional bases that are related to the barcodes. So, because obviously when you assemble your DNA, that's not part of the genome and you need to get rid of that and any adapters or anything that is attached to your actual DNA. And the program that is generally used is Guppy. There are others, as I say, but Guppy is an Oxford Nanopore program, and it's what Minnow uses as well. So it's quite easy to use, I think. And that you can then go and assess that you can use to do that. And I'm going to show you examples of FastQC and multi-QC. You can then assemble all of your FastQ files. So FastQ files, as I say, are sort of collections of roughly about 4,000 bases, or 4,000 reads rather, and each of those will be a different amount of bases. And you need to assemble those into one sort of large genome, or at least contigs, which are sort of collections of sections of the genome. And the way you can do this is either compare it to a reference genome if you already know what your isolate is. So if you know that you're sequencing a culture of TB, then you can compare it easier because you have a sort of guideline to do that for. Or if you don't know what the isolate is, or it's a relatively new thing that doesn't have very many reference genomes available, you can also do de novo assembly, which is when you basically just kind of like doing a jigsaw puzzle blind a little bit. You have to just piece the pieces together without very much information. And there are different programs that you can use to do either of these things. And again, there are lots of other programs that you could also use. But I believe Minimap is a nanopore one. I'm not sure about Fly. I don't think that's a nanopore one, but I found they both work quite well. You can then polish the genome. So that's just going over and making sure that what you think is the right base at each point is what you think it is. This is quite important with nanopore sequencing because nanopore sequencing is not as accurate as Illumina. And so going over and just checking everything is very worthwhile. And a program called Medaka is quite useful for doing that. And then once you've got your either whole genome assembled or you've got your sort of contigs assembled, you can then do downstream analysis in the first place. So creating a phylogenetic tree of the genes and sort of different bits of information on it, or doing variant calling, looking for SNPs, looking for antimicrobial resistance genes, you know, whatever your aim was in the first place. Once you've got your assembly, then the world is your oyster. So that's kind of the basic gist of what we're going to look at. I'm going to explain more this sort of side, the base calling, quality control, and then the assembly. And then obviously the downstream analysis is more your choice of what you want to do with it. So I'll explain a little bit about them, but I won't go through them in too much detail. We can now look at the common file types you might come into contact with during your processing. So at the base calling stage, as I said, the raw files are these fast5 files, and you'll see the file extension will be .fast5. And these are the direct output from sequencing. So these are the squiggle data, so the changes in the electrical current that is created when it goes through the pore. And they store a huge amount of information. And if anybody's ever had a look at the raw data, you'll see that it's gigabytes worth of data. So they are really quite large. You can't open them with a text, excuse me, with a text editor. And this picture, I couldn't really find obviously a picture of one, but basically what it is it's collecting this information here about the different squiggles as the pore, as the bases go through the pore. We've then, we've then got FASTQ files. So this is what FAST5 files are changed into when base calling occurs. So these are the A's, the T's, the G's, and the C's. And the file extension is .fastq. And FASTQ is basically the faster files plus quality scores. And they can have multiple sequences or reads within the files. So FASTQ files typically, as a sort of general setting, store about, I think it's 4,000 different reads within each file. As I said, reads will be differing lengths depending on what sequencing you've done, but so they can store quite a lot of information. And each sequence read is defined in these four different lines. I won't go too deep into it because I've put information on the PDF, but you can see that here's a read, and then there's another read here. So Sylvia very kindly provided this for me. And then you've got the information regarding those reads, so the quality scores, the information surrounding it. We've also got faster files, so the file extension .fna. This is very much just the A's, T's, G's, and C's, and they're often used in reference database within the file, and it's defined as a header and then the sequence code. So you can see down here we've got TB. Yeah. There's also SAM or BAM files, so sequence alignment map files and binary alignment map files. These you'll come across when you have aligned against a reference sequence. So these are from sort of Minimap, and they start with a header again, followed by the alignment information, notepad or a text reader or something like that. So you can actually physically see the information that is within them. We've then got VCF, or variant calling format. So this is when, if you're doing, if you're looking for SNPs or single insertions and deletions, if you compare it to, if you compare your genome to another genome, what you'll get is, again, you can read it in Notepad, and it will provide the base points. So as you can see down here in this red box, your reference genome, it says it's a T, but the alternative, which is your genome, suggests something different. So it basically goes along and picks out all of the SNPs that are different and points out what the difference is. And these can be quite large files. So you might find that there's quite a lot of SNPs or individual bases, so they're quite large. Also, other file types that you might come across as well in GeneBank, the .gbff and GFF are used for annotated genomes, so you can download those off the GeneBank website, and you can open them in something like Artemis, and you can see all of the individual annotated genes. And then there are other different formats. Obviously, things like phylogenetic tree programs utilize loads of different file names, file types, for instance, .tree. And the more, obviously, you get into this, the more you will see that there are different extensions. We can then move on to basic code structure. So I just wanted to explain, hopefully, roughly how code is generally structured. It generally starts with the program. P has a couple of different sort of programs within it. So Ducky _ base caller is the program that you will use to change FAST5 files to FASTQ files. You've then got the command, which is the action that you want that program to do. And then sort of below that, you've got arguments and parameters, flags and options, and all of these are kind of there to explain to the program within that command, right, I want you to do this, but I want you to do this in a certain way to a certain file or to a certain path, and just sort of specify the options. So on most of so GitHub or on the website, on the web page that is sort of attached to a program, usually you'll find that there's flags, options, and it will explain what each one does. And they're really useful when you're looking at how to sort of use a program for the first time. And a few other points I have found, and I suspect everybody else finds, it's really, really useful to write down your code and annotate it so you know exactly what it is and that you can come back to it later. I use Notepad. Some people use Notepad++, which I think color codes the code, or Xcode if you use Mac OS. And it's a really useful way of coming back to it. So I have a folder full of Notepad files that explain exactly what it is. So not only do I have the specific code, but I also have lines above it that start with a hashtag and that explain to me exactly what this code is for, use it, and what each bit means, because it means you come back to different sort of input or output. And it's I thoroughly recommend that, because you might come back to something after 6 months and not have any idea what it is. So yeah, the more you annotate it, the better, definitely. You can also pipe different commands together so that they follow on from each other. And the pipe symbol is the long straight line here. And you can also address tools and programs and save them within a variable within the environment that you're working in, which allows you to just call the tool by name instead of providing the entire path. So I'll kind of show you. But within Linux, there's two different ways of doing it. You can have a hard link or a soft link. And a hard link the link to that program basically stays where it is, even if you move the file around within your system. And the soft link is more like on your Windows desktop. It saves time. Everybody's favorite, dealing with errors. Errors are really common, especially when you're learning, and don't let it put you off. They're sometimes quite hard to identify, but somebody will know how to fix it. Something useful to know, though, is that commands are executed from the start of the line to the finish of the line. So wherever it encounters a problem, everything before that will have worked. So for instance, if there's a problem with the first part or it can't find the program, it will stop the command before it's done any of the rest of it. However, if, for instance, your error says, I can't find the folder that you want to output to, it suggests that everything else before that is fine, and it's got to that point and it doesn't know where to put it. So commands, errors usually are quite helpful, and they are designed to help you identify what the problem is. Sometimes you might not necessarily understand what it means, but then error log should be generated, which means you can open it and look into it. And the internet is a really helpful place for identifying errors and trying to fix them. There are lots of different forums for this kind of thing. Something called Stack Overflow and BioStar are both really, really helpful. You can either paste in the error or type in what your problem is. And usually, I almost guarantee that somebody will have had that problem before. So take your time and have a look through those forums, or just Google it. I have lost count of the number of things that I have failed to do correctly, and then just Google the problem. And you can copy and paste the actual error message, and that can help sort of identify what the problem is. So check the internet. There are lots of different people online who definitely will have had this problem before or know how to deal with it. And don't worry, don't panic. I just wanted to talk a little bit about Anaconda. Anaconda is a program that enables you to install software on your computer, and it basically installs all of the existing programs that you need for that program. So for instance, Medaka uses programs from Guppy, so it makes sure that you have literally update it, and it updates everything. So it helps to bring everything kind of together, and it stops you from installing one thing and not having the correct version of another thing that you didn't necessarily even know you needed. And I know that there are other versions of other ways to do this, such as Docker, but I've explained a bit more in the PDF about this, but I've certainly found that Anaconda is quite useful for installing programs and making sure you've got everything that you need.